The Division of Translational and Regenerative Medicine is a new division to focus on research within the clinical department, the Department of Medicine, and the University of Arizona College of Medicine. We have three missions. One is to conduct basic translational, clinical, and population health sciences research to understand human disease, more specifically on respiratory diseases and cancer. And secondly is to provide infrastructure and co-facilities for enhancing collaborative research so we can increase the research productivity. And the third mission is to provide an academic environment to train clinical fellows to conduct basic and translational research, but also to train PhD investigators to conduct disease-oriented translational research.
We have been able to drive pathways that allows us to develop potentially therapeutic interventions for the diseases that we're studying in the lung. And so by moving here, that actually gave us the access to these other investigators that actually allow us to pursue this in a more translational, potentially clinical arena.
So the focus of my research is really understanding the molecular underpinnings of acute lung injury or acute respiratory distress syndrome. And we're kind of expanding that into also understanding lung cancer and a variety of other lung diseases. A primary area of research in my lab is understanding redox regulation and its role in not only cell physiology and cell function, but also how it may become altered in disease states. We're interested in understanding that whole process and how it gets dysregulated within the setting of oxidative stress in particular. So another focal area in my lab is the receptor for advanced glycation end product, which is a pattern recognition receptor as part of the immunoglobulin superfamily. So we've been working to better understand what its role is in physiology so we can understand how it might be involved in pathology.
My main focus in my research is right ventricular function and right heart failure. So I take a cardiovascular approach to it, or a biomedical engineering approach to it, of looking at the cardiovascular hemodynamics. How does the blood flow change in disease when there's structural remodeling in the pulmonary vasculature as well as changes in the right ventricle? So I approach that by investigating this in patients with pulmonary hypertension as well as animal models of pulmonary hypertension. The animal models that I've helped work on and develop are actually associated with pulmonary hypertension due to left heart disease. As you have this dysfunction in the left ventricle and you have this buildup of pressure, is there a component of pulmonary vascular disease or remodeling that contributes to this pulmonary hypertension? It's called pulmonary hypertension in the setting of heart failure preserved ejection fraction. I helped develop phenotype the animals in these models.
The main research focus in my lab is the pathogenic mechanisms and the therapeutic interventions of the idiopathic pulmonary arterial hypertension. Most specifically, we are interested in membrane receptors and ion channels and calcium signaling and their pathogenic role in the development and progression of this disease. So our approach is that if you can reduce the pulmonary vascular resistance, or if you can reduce pulmonary arterial pressure using different drugs, you can relieve the increased afterload for the RV or right ventricles, and then relieve the sort of burden for the right ventricle, and maybe there will be an efficient way to treat right heart failure.
The focus of my research is actually on endothelial cell biology. We've been focusing on the role of the mitochondria. What we found is that in many diseases, at least that we're interested in the lung, is that the mitochondria becomes dysfunctional. And when it becomes dysfunctional, it has huge impacts in terms of the cell biology. And we've shown that if we give them one of the substrates for that, called carnitine, it will restore their beta oxidation, and the nitric oxide function actually is restored. And so their blood vessels actually will vasodilate normally. One of the other things that I work on is actually acute lung injury. And so we've identified a very specific event on one amino acid in that protein, and it's called a tyrosine nitration. And so what we have identified is these things that we call nippers. So they're nitration inhibitors of the Rho GTPases. And then by doing that, we could shield that particular amino acid. And what we find is that when we put that into animals, is that the mice don't get acute lung injury. So we're very, very excited about that.
It's a very high quality team taking an in-depth look into the research.
And that's one of the cool things about the division is that we have great teams because we have such great individuals.
So we have some very heavy hitters here that do some outstanding, groundbreaking, moving the science forward type of work.
So our division sort of like tries to provide a good environment and infrastructure and academic environment to train physicians so that they know how to do research, they know how to design experiments, and eventually probably can develop their own academic career by doing different kind of research on something they're interested in doing in the future.